Hi guys, this is Ruben again, and this is the third video blog for the BSPP, and today I'm going to talk about some of the experiments that I carried out during my stay at the GAC. So first of all, we carried out an antagonism assay to determine if we could mix the selected biocontrol candidates in synthetic consortia. These are the selected biocontrols that I mentioned in the previous videos, and for that we pipetted a drop of an overnight culture of one of the strains on top of a water overlay containing other of the candidates. As you can see in the pictures, these were some of the results. So after that, we transformed the pathogen PSA with a plasmid that provided tetracycline resistance to selectively recover it after the following experiment. So the following experiment was the implant assay, inoculating groups of 12 tomato plants with overnight cultures of biocontrols and PSA. We sprayed the plants with groups of the selective consortia plus PSA, isolated biocontrols plus PSA, a negative control with just water, and a positive control with the pathogen. Then we collected leaf samples on days 1, 3, and 7, and after some processing, we pipetted some of the dilutions in the media, in KB media, and after 2 days of growth, we compared the colony forming units of the pathogen in the different combinations with a positive control to assess which biocontrols and consortia were effective when inhibiting PSA, and also if the biocontrols work better isolated or when working in groups. Finally, we also performed a technique called transposon mutagenesis over the biocontrol candidates using transposon TM5, and this is a technique to generate libraries of phenotypes of interest, and in our case the phenotypes of interest were loss of PSA inhibition, loss of streptomycin casugamycin resistance, and loss of copper resistance. And the last two ones were to ensure environmental safety and not to spread new antimicrobial or antibiotic resistances into the nature in case we might have discovered new genetic determinants that could be spread into other bacteria in the plants. So in the next video, I'll be talking about some of the results that we obtained with this experiment. So see you all there.